Good afternoon everyone. Today I am going to teach you regarding the Unit 4 of Pharmaceutical Biotechnology. So in this Unit 4 we have the immunoblotting techniques and the microbial genetics. The immunoblotting techniques, it will include the Western blotting, Southern blotting, ELISA techniques, and genetic organization. It will be including the eukaryote genetic organization, prokaryotic genetic organization, and also it will be including the biotransformation and the mutation part. So we will start with the immunoblotting techniques. Immunoblotting techniques are used to detect the specific proteins or nucleic acids based on antigen and antibody interaction or nucleic acid hybridization. Major techniques are ELISA, Western blotting, and Southern blotting. So ELISA, it is also known as the enzyme-linked immunosorbent assay. It is very sensitive immunological assay used to detect antigen or antibody based on enzyme-linked antibodies producing measurable color changes. And different types of the ELISA techniques are available: direct, indirect, sandwich, and competitive ELISA techniques. So applications of these techniques are disease diagnosis, vaccine evaluation, protein quantification. So here it is a small chart for the process of ELISA, the principle. So antigen will be coated in the well plates, and then primary antibodies will be applied above this, and then finally it will be detected with the enzyme linked antibody. So these are the different types and how the normal ELISA we can conduct with. So the target antigen bind to capture the antibodies will be placed on the surface of the plate, the 96 well plate, and then the primary antibodies will be attached with this antigen, and then finally we will be attaching the substrate, that is the enzyme, which will be detecting the signal of the reaction. Western blotting technique is used to detect specific proteins in a sample. So here we are using the SDS-based gel, which will be transferred into a membrane, and with the antibody detection we can find out that the antigen is present or not. So the primary antibody binds to the target protein, and secondary antibodies will be binding with the enzyme, which will produce a detectable signal, and we can able to find out a specific or targeted protein. So the flow chart of this Western blotting technique in short. So protein sample will be analyzed or run through the gel electrophoresis, then it will transfer through the membrane transfer, and then finally it will be detected with the antibody. So here is the structure with more clarification. So denaturation of the total protein will be loaded with the loading dye into the gel electrophoresis, and here the gel we used to run in a system, and then we used to get the protein into the gel. We try to transfer it into the membrane, and finally it will transfer into the membrane, and we will be treating it with the primary antibody, and then we will be incubating it with the secondary antibody, which will be linked with an enzyme, and finally we can detect the presence of the antigen or the protein. So Southern blotting, it is used for the DNA sequences detection, and DNA fragments separated by agarose gel electrophoresis. So it will be transferred into a membrane, and then again the leveling will be done with the enzyme, and finally the applications we can get is the the gene identification, mutation detection, and DNA fingerprinting. So this is the structural representation of the Southern blotting technique, and here the genetic genomic DNA, we are just making it into small cuts with the restriction endonuclease enzyme, and then we are running into agarose gel electrophoresis. The DNA segments, it will be getting segregated in the gel electrophoresis, and finally we are transferring into the nitrocellulose membrane, and we are going to treat it with the antibodies to get the specific presence of the DNA. So genetic organization of prokaryotes, it will be a single circular chromosome, DNA located in nucleoid origin, and genes arranged in operons, presence of plasmids carrying extra genes. So here the structure, it is showing here the nucleoid region where the supercoiling of the DNA material, it is there, and DNA, it will be forming in a loop structure. So it is a very simplified genomic organization in the prokaryotic cells. And genomic organization of eukaryotes, here it will be multiple linear chromosomes were present, DNA enclosed inside nucleus, contains introns and exons, and complex gene regulation mechanisms are involved. So here it will be more complicated. So here with the DNA helix, it will be the presence of the chromatin where this DNA helix will be wrapped out, and then it will be forming the whole set of chromosomes. Microbial genetics. So here it will be including 3 different processes of the microbial genetics. So here the microbes are involved in the transfer of the genetic material, and here transformation in the transduction and conjugation. These are the 3 basic genetic transfer methods it is available. So genetic elements include plasmids and transposons also we have to study in the microbial genetics. So first one is the transformation, uptake of free DNA fragments from the environment, DNA incorporated into bacterial genome, an important tool in genetic engineering. So here it is a simple structure of the bacterial transformation. So the foreign genetic material, it is around the environment, it will be taken by the bacterial cell, and they will incorporate it in their own chromosome, and the gene will be transferred from one place to another. Next process is the transduction and transfer of DNA via bacteriophages. 2 types, generalized and specialized transduction, important in horizontal gene transfer. So here also one simplified figure it is presented here. So the bacteriophage are involved here, which will be transferring their genetic material into the infected bacteria. The genetic material, it will enter into the bacterial cell, then they will be producing the viral genetic material inside the bacterial cell, and after that they will be lysing the bacterial cell, and they are coming out, and finally they are infecting the another new bacterial cell, and it will be transferring the genetic material from one bacteria to another bacteria with the bacteriophage. So conjugation is another one method where transfer of genetic material through direct cell to cell contact, and it involves the F plasmid and sex pillars, important mechanism for antibiotic resistance transfer. So here this is a simplified diagram of donor and recipient bacterial cells, where the donor cell used to make a pillar structure with the recipient bacterial cell, and they used to transfer the genetic material from the donor cell to the recipient cell, and finally both the donor cell and recipient cell will be having one copy of the genetic material. So these are the different methods of genetic transfer in the microorganism. So next one is the plasmids. Small circular extrachromosomal DNA molecules replicate independently of chromosome, carry genes for antibiotic resistance or metabolic functions. So this is the simplified structure of the plasmid. Here it is representing, it will be having origin of replication, antibiotic resistant gene, and different sites for the primer to bind, and promoters also, and selectable markers also. So restriction sites are available here, where the restriction endonucleases can cut the plasmid and we can insert a foreign gene sequence into the plasmid to transfer. Transposons are also known as jumping genes, mobile genetic elements that move within genome, cause gene rearrangements and mutations, important in genome evaluation. So this transposon structure, here it is explained here. So the donor DNA will be having the transposon, and the recipient DNA, it will not having before the transposon sequence, but they have this the transposon, it used to jump from the donor DNA to the recipient DNA with the help of one enzyme that is called as a transposase enzyme, and they used to cut the recipient gene structure and they used to install the transposon structure into that, and from the donor DNA they used to jump into the recipient DNA where they want to target. So this is about the transposon genes. Then microbial biotransformation, chemical modification of compounds by microorganisms used in drug metabolism studies, and applications are steroid transformation, antibiotic synthesis, and bioremediation. So this is a specifically very simple structure of presenting the biotransformation. Here bacteria and or fungi, they have they used to go through the fermentation process, and the parental compound it is present there, it will be changing from the fermentation process, and it used to go for biotransformation. Then we used to extract the biotransformed product, and then finally we will go for the analysis of through the chromatography, then NMR study, and finally we used to find out a biotransformed product or compound which will be having different biological activities like anti-cancer, antimicrobial, neuroprotective, or anti-parasitic activities. So it can be any activities, but here I just presented a specific activities, so it can be many different other activities also. So mutation is a permanent change in DNA sequence, can occur naturally or be induced by mutagens, important source of genetic variation. So different types of the mutations are there: point mutation, insertion mutation, deletion mutation, frame shift mutation. So different types of mutation here it is mentioned in this figure. Substitutions, deletions, insertion, inversions. So here with this diagram in the substitution, we are substituting one or two more sequences with the other new sequences, so that is called substitution. Deletion means one or more DNA sequence we are just deleting from the whole sequence, so that is the deletion. Insertion means on a new specific gene sequence we are inserting into the DNA segment, so that is called insertion. And inversion means we are just shifting the arrangement of the genetic base pairs, so that is called the inversion. So these are the different types of mutations. I hope everyone will understand, and it is very simple also. Thank you so much for attending the course. Thank you.